Good afternoon, everyone. Thank you so much for coming, and I understand that you are joining me directly after lunch, so I will not be offended if the cool blue images of shipwrecks lull you into a post-lunch coma. So this is a work in progress. Please bear with me. I'm still awaiting definitive results. So if people have any suggestions on different methods that I can use to get better results out of the various materials I'm looking at, please let me know. So there have been several studies that have sought to analyze ancient DNA from underwater archaeological sites. People have looked at waterlogged grape seeds. They have looked at desiccated and waterlogged olive pits. Even human remains have been analyzed to look for tuberculosis bacterium. However, the ones that interested me the most as an underwater archaeologist who primarily excavates ancient shipwrecks were two studies, one published in 2008 and one published in 2012, that claimed you could get genuinely ancient DNA out of the ceramic matrix of amphora, or ceramic transport vessels. Now, what made me so curious about this was the fact that the majority of ancient shipwrecks, as you can see in this image here from the Mosotos shipwreck in Cyprus, what we really find on them are ceramic cargoes. So if we had a way to characterize the past contents of these vessels, then it would greatly illuminate the shipwrecks that I work on and that I study. However, these past studies, they all have several things in common, which I found a bit worrying. They all utilized polymerase chain reaction as opposed to next generation sequencing, which we now have available to us. And by doing this, they only amplified pre-selected DNA. So obviously in the case of the olive stones, they were just interested in looking at olive DNA. In the case of the grape pips, grape DNA, humans, tuberculosis. And for the ceramic matrix, what I found especially worrying was that only food items that they expected to find were amplified. And this was even made explicitly clear by the authors, where they said that they designed primers to amplify things that were, quote, part of a typical Greek diet. And in addition to this, none of these studies took the time to test the deposition environment. In this case, in underwater, it would be the seafloor sediment and the water column itself. So back to what these amphora contain. Perhaps the reason why these authors were so comfortable looking at typical diet things is that I find when you look at amphora studies generally, and especially on ancient shipwrecks, we generally sort of assume we know what these vessels contained. Primarily olive oil, wine, or fish sauce. However, due to the preservation of organic material in the occasionally anoxic environment of seafloor sediments, what macro remains actually show is that there was a huge variety of things that were contained in ancient amphora, including resin, as found on the Uluburun shipwreck. There are fig seeds that have been found in numerous amphora. On the Kutaş shipwreck found off the coast of Turkey, in this pseudo-Samian wine amphora, remember, wine amphora, they actually found butchered beef bones. Now, what's really important about this point is that butchered beef was not thought to be a typical element of the Greek diet. So if you had only amplified pre-selected DNA, you never in a million years would have thought, well, let's amplify bovine DNA. On the Kyrenia shipwreck, we found whole almonds. I have worked on wrecks where we found thousands and thousands of olive stones inside of what were thought to be wine amphora. We got pomegranates. The list goes on and on. When you actually look at the macro remains, you find there's much more multifarious uses of amphora, and their uses were nuanced, and it just goes on and on. So the questions I'm really interested in answering are the following. Does genuinely ancient DNA survive inside of the ceramic matrix? And what DNA is present in the deposition environment? And how could DNA that's just naturally present in the deposition environment affect our results if we try to deduce the past contents of vessels from ancient shipwrecks? There's also have been claimed that due to the very consistent temperatures you find on very deep shipwrecks and the lack of UV sunlight, that ancient DNA is more likely to survive in underwater contexts than on terrestrial contexts. I would love that to be true as an underwater archaeologist, but we need to investigate this further to give definitive answers to questions like that. And in addition, I know that this is a very, very fast-moving field, so now we're lucky enough to have powerful tools like next-generation sequencing and metagenomic data sets that we can look at, as opposed to only amplifying DNA that we're hoping to find in the first place. In addition, I will be doing a tandem study, it's currently underway, where we look at what results we actually get from these ceramic vessels through traditional residue analysis, through gas chromatography, mass spectrometry. So my data looks at 6 different ancient shipwreck sites from a large variety of time periods, from the late Bronze Age to the late Roman Empire. Here's a map of where my sites are. So as you can see, this only spans the Mediterranean at this point. I actually just returned from Malta at midnight last night, and I've been working with a bunch of colleagues who work in the Baltic, and very excitingly, it looks like my data set might be able to be expanded from beyond just the Mediterranean into the Baltic and other bodies of water that have very different conditions. So we're hoping that this map can be greatly expanded. Furthermore, some of the work that was done previously claimed, very importantly, that you could do non-destructive analysis. As I'm sure you can all appreciate, the most difficult aspect of what we do on a lot of levels is getting curators and principal investigators to give us permission to do destructive analysis. Now, this 2012 paper that came out that claimed you could deduce past vessel contents simply by swabbing the insides of these amphora, I thought if that was true, that would be revolutionary. If you could imagine being able to go into the British Museum or the National Museums here and simply swab ceramic vessels and tell definitively what was inside of them, that would be absolutely incredible. But we need to do more research to determine if this is actually the case. I myself am quite skeptical about this. So another interesting aspect I find with my work that differed from these past studies was I was lucky enough through my various work and contacts in underwater archaeology to start from a known quantity. So I have deliberately selected a number of vessels of known contents. So from the Uluburun shipwreck, the late Bronze Age wreck, I have tested a number of vessels that we know contained resin, Pistacia terebinthus resin, which was used to make perfumes and unguents in the ancient world. In addition, I'm testing vessels that were known to contain whole olives and vessels that were known to contain whole almonds. So it will be really interesting to see if any DNA from what we know the vessels contained is inside of there. Furthermore, I like to challenge these assumptions that I brought up earlier that vessels only contained wine or olive oil or fish sauce. So another aspect of my research is I have deliberately selected vessels that have assumed contents. So in the case of the Kyrenia shipwreck, Rhodian amphora are always assumed to have contained wine. We all know that Rhodes had a massive wine industry, but I for one believe that they're produced a variety of agricultural products, and so we can't be sure definitively what every single one of these vessels contained. In addition, the Babujas shipwreck in Croatia contained key 25/Africana-3 amphora, which are always thought to contain olive oil. So, do we see any DNA preserved from these assumed contents or not? In terms of sample procurements, I actually have to go to these lovely countries. Obviously, this is the worst part of my job, is going to places like Cyprus and Croatia to get them for destructive analysis, where I actually get to bring the sherds back to Oxford and Warwick and York. In terms of the non-destructive analysis, it's a simple case of swabbing them using the same methods that were described in the 2012 paper to see if we can replicate those results. The analysis I'm undertaking is occurring at the DNA side at the University of Warwick with my lovely colleagues from Warwick who are here, and the University of York has been collaborating with me on my residue analysis in a tandem study. Now, the variety of shipwrecks I think is really fun because we're looking at a vast expanse of time and also different depths. So for example, the Uluburun shipwreck sits at 43 meters, which might have a bit more of a stable environment, more of a stable temperature, and be less likely to be affected by wind action and wave action, which would disrupt much shallower shipwrecks. And as you can see from this little selection of the kind of artifacts that exist on the Uluburun, there would, if the DNA survives, be the potential for lots of different kinds of DNA. In terms of just animal remains alone, there was hippopotamus ivory, elephant ivory, and a variety of other goods like astragals, knuckle bones from a variety of animals that we think were used as gaming pieces, in addition to organic cargoes like the Pistacia terebinthus resin that you can see here. Now, I just returned last night from a recently discovered shipwreck in Malta, the Jlendi wreck, which I am really excited about because this is a Phoenician wreck, and we know for a fact that the Phoenicians were trading from Spain to Syria, and in a lot of levels in the Iron Age, they dominated seafaring trade, and so it's doubtless that they traded in a variety of goods. So this will be really exciting to see what we find from there. The Mazotos shipwreck, I think, is one of these perfect examples of how we need to look at assumptions we make. The variety of the cargo here were Chian amphora, always thought to contain wine, but when I was working on the project in 2012, I brought up this amphora, which as you can see contained 2,000 olive stones, these preserved olives. So let's not assume that we can just amplify DNA from what we expect to find. In this case, it would have been grape DNA. The same could be said for the Kyrenia shipwreck, where we found thousands and thousands of almonds on the wreck that were included in the Rhodian amphora. And again, these Rhodian amphora are always assumed to contain wine, but let's challenge these assumptions with DNA. Again, with the Bobulia shipwreck, what people thought always contained olive oil, a study by some of my colleagues from Oxford and others that came out in 2015 showed that through residue analysis, it seems more likely that the ones they tested contained sesame oil as opposed to olive oil. Now, I did include one site in my studies, a terrestrial site, just to compare if we can look at the preservation of DNA from a terrestrial site versus underwater sites, and whether or not those assumptions that DNA is more likely to survive underwater than on land can be tested. Now, I am really concerned about lots of sources of potential contamination. Even when we bring artifacts up from the seafloor, often we bring them up not in containers, as you can see me bringing this basket of artifacts up in Croatia, and I'm really concerned about the different kinds of DNA that might be in the water column itself. For that reason, I take water column samples from every shipwreck that I work on and would like to compare the DNA that I can amplify and study from the water column to what we get from the ceramics and the artifacts. In terms of potential sources of contamination, ancient shipwrecks are notorious for becoming new artificial reefs. So as you can see from this photo, there's a little eel that lives inside of that amphora. And here's a little starfish that I saved from going up the dredge and perishing in dramatic fashion. So I would be absolutely shocked if we don't end up getting at least some eel or octopus or starfish DNA from some of these amphora. But of course, we can't expect those to have been the original contents. And this little guy that I just brought up 2 days ago in Malta, as you can see, there's a very nasty sea urchin in there, so we have to anticipate that in the very dynamic marine environment, we will encounter loads of different kinds of DNA. In addition to my non-destructive ones, and this is no criticism, by the way, on my colleagues at the Institute of Nautical Archaeology in Turkey, the samples, some of the actual ceramic vessels were stored 10 feet from where this photograph was taken. And I don't know about you guys, but can anyone see any potential sources of DNA contamination in here? For example, that's actually a grape plant that's growing up right there. So if we think we're amplifying grape DNA and it's genuinely ancient, we need to be really careful about that. In addition, I'm kind of hoping that it might look like these vessels contained cats. And just to sum up, it's really important that first we characterize the deposition environment. I can't stress that enough. In addition, I'm really curious about what kind of marine biology information we can get from this, because the marine environment is hugely dynamic, and these ancient shipwrecks are artificial reefs, so I fully anticipate there will be lots of really interesting things. In addition, DNA found in a museum storage environment, if you're looking at doing non-destructive samples, we have to be really clear about what DNA exists in just the dusts and particles that you're going to find in these environments first, before we assume that it has anything to do with past vessel contents. This is ongoing work, so any suggestions or critiques that you could lend me, I really appreciate. We'll obviously be looking at things like map damage and base pair fragment length to try to determine if this is genuinely ancient or modern. And I'm really excited about comparing these results to to the results from my residue analysis to see if there's anything that ancient DNA could show us that the residue analysis results are incapable of. Thank you so much for listening, and yeah.
Thank you very much. I think we have time for a short question.
Have you seen any vessels where DNA both like wide and out with DNA? Because like maybe bricks to the nose, like in layers like all water. So like 75 years ago.
Yeah, the use and reuse of amphora is of primary concern to me. Because I think there's a variety of scholars, especially, I don't know if you know Ted Pena, Theodore Pena has looked at the use and reuse of ceramic vessels, and it seems to be without a shadow of a doubt that they were frequently reused. So if anything, another thing we'll have to be looking at is if we do find residues or DNA, does it represent the first, last, or middle use of a vessel? That's absolutely.
Thank you very much. So let's thank Lisa again. Where about the talk about marine
Thank you.
So for those of you who have not asked to meet yet, I'm a postdoc at Robin Alvey's lab at the University of Warwick, and I'm also a member of the Lost Frontiers project. So that's a big ERC grant that's looking at using seismic mapping, sedimentary DNA, environmental analysis, and agent-based modeling to look at Doggerland, this area here. This area here, which was the land between England and France. It formed a fertile, habitable land prior to its inundation about 8.2 to 8.5 thousand years ago. And as part of the project, we're looking to see if we can reconstruct the past environment, to look at environmental change, and to look for the signatures of human habitation. That's really the key point of the project. Is really we're wanting to focus down, see if we can detect patterns of human habitation. So we're about halfway through the project. It's a 5-year project, just a little over halfway. But I wanted to give you some of the preliminary results that we can see. And I'm focusing on the sedimentary DNA today. That's my area of expertise. Okay, so from the 72 cores we've retrieved so far from the North Sea, we've got a broad variety of different environment types represented. So we see reef swamps, we see shelly deposits, paleosols, lacustine deposits. We see peat layers from a couple of centimetres thick to up to 1.8 metres of peat. We see beautifully laminated intertidal muds. But I want to talk just about the first sort of tranche of cores that we took. So this is focusing down into the just off the wash of the east coast of England. So from the first 14 cores, we took 92 samples for cell-DNA analysis. And that's shown here by each of the squares. So one square is one sediment sample. And these are very, very preliminary results. They do show some of we subject them to low coverage metagenomic sequencing, and they show some of the characteristic patterns of damage that we expect to see from ancient DNA samples of this type. But there's not much we can say, there's a lot of masses of data. But we can, here I'm just looking at the plants, and we can probably tell between fully terrestrial environments and more aquatic, increasingly aquatic environments. Just to reiterate again, they're not subjected to our, whilst we see broad patterns of damage, they are subjected to our sort of rigorous phylogenetic intersection analysis, which I'll talk a bit more about later. But they give us an idea of which cores are going to be useful to us, which have the best DNA preservation, and which ones might be the most interesting from a set of DNA perspective, but also from all the other proxies that we're looking at. But before I really want to talk about that, I want to talk a little bit about the effect of seawater on DNA. So as we know, water is a polar molecule, and the primary processes of ancient DNA damage are through hydrolysis of the DNA molecule facilitated by the polarity of water. And previous studies have showed that in highly ionic strength environments, such as marine environments, there's a dampening effect of the impact of the polarity of water. We see lower levels of cytosine deamination and also fragmentation, where we have high concentration of salt. So what we wanted to do is we wanted to start off with some experiments to see if we could replicate that in the lab. And we took model DNA and incubated it for to different concentrations for different temperatures. So we start with in water, low iron strength salts, and higher iron strength salts. And 0.6 molar is the same iron strength you'd expect to find in seawater. And what we found was that at 4 degrees, down here, which is the maximum density of water, so that density when the water is at 4 degrees, it sinks to the bottom, to the temperature of the bottom of the ocean, and lots of sea floor sediments tends to be about 4 degrees. And what we found was that at 4 degrees and in a high ion strength environment, the rate of cytosine deamination will be reduced up to 12-fold when compared to water. Even in warmer temperatures, at 37 degrees, for example, we saw a 5 to 6-fold reduction in the level of cytosine deamination, which is really interesting. But just to reiterate, these are free DNA in solution. So what we're going to do next is we're going to replicate some of these experiments, but look at some of the impacts of different sediment types and see if we can see how the interactions of sediment and DNA and different types of salts will impact these findings. Okay, so to talk briefly about work we published in 2017, which then took the then available 185 paleogenic data sets in order to agree calibrate some of our models of expectations of DNA damage. And what we saw then was that while cytosine deamination is largely a thermal age process, fragmentation, so lambda, which is our usual measure of fragmentation, is not it doesn't have an age-dependent process associated with it. And so we find it as a better marker of environment than cytosine deamination. So going back to our preliminary data from our sediment cores, we found we were looking at lambda here, so that's the measure of fragmentation. And we looked at different environment types. So we broadly categorized different types of sediment that we've got here. That's using the DNA data, but also the other proxies we used. And what we found was that in saline environments, the level of damage, so small and low damage, in saline environments, the level of damage is significantly reduced. Generally, if you've got a saline environment, your DNA is likely to show less damage than you might expect from, say, a shorter environment. That's what we see. But that's just preliminary data. So I just want to go on to some of the findings, take that into this one particular core, which really stood out to us from the start. It's core number 1A. And this is a better picture of it. So this is the top of the core, and it doesn't show it particularly well. At the top of the core over here, the bottom over there, the top part of the core is mainly cephalodon sands and gravels, but from around about here downwards, what we see is a really beautiful sequence of laminated fine sands and silts, except for here, which is where we see this sort of about 20 centimetres of massive unconsolidated, shelly gravel, lots of broken shells. Which still appears to be sort of out of place in the sequence. And with that, we see some unusual chemical signatures. So here I'm just showing the sulfur levels. We see a marked spike at this stratum. And when we look at the diatom complexity, so this is below the deposit, we see a really high complex, there's really great retrieval of diatoms. During and after our deposit, we virtually see no diatoms in there. What is there? There's not really enough even to put up on these plots. It's a real reduction, a marked reduction in the complexity of the diatoms that are there. So something's happening there. So interesting is definitely happening. And we have put fairly preliminary dates on this deposit to about 8.2 thousand years ago using OSL dating. So where does that fit in with the DNA? So this is our preliminary data. Our unusual stratum does show some damage. That's expected with samples of an ancient DNA, but it's not so damaged as to be unworkable. So what we did is we took 5 samples, 2 below, 1 during, and 2 above the deposit, and subjected them to high throughput metagenomic analysis, much higher coverage than we had previously. So I'm not going to dwell on the details of this figure in the interest of time. If you want to come talk to me about it later, that would be great. But just to sort of show, what I want to highlight really is that we've got a real shift in the bacterial communities as you progress along the core. So these two here represent the laminated intertidal worlds before the deposit. This one here is our deposit, and this is after our deposit. And what we see, particularly in our deposit, we see a particularly marked and distinct community with a real shift towards marine-associated taxa. And indeed, we were able to recover a low-coverage genome of Biomolecular Archaeology, which is a deep-sea hydrothermal vent-associated bacterium. And just to point out, from this point onwards, all the data has been subjected to our phylogenetic intersection analysis to make sure that we really can be confident in the assignations that we're giving to our data. I'm not going to go, I don't have time to go into the details of how that works, but if you want to know more about that, please come and have a chat with me. Becky Crimpton in the audience had a poster yesterday, so come and have a chat with her as well if you didn't manage to see the poster yesterday. But yes, just to go on now. And to give you an idea of the stringency that we use in our data assignment, around 3% of the reads that we sequence have a match in the database. So shotgun sequencing is pretty standard. And that's not because